Last time, we followed electricity through the heart like a route on a map. SA node, AV node, bundle branches, Purkinje fibers. We knew where it went. This time, we ask a different question. What is that electricity? Where does it come from? To answer this question, we need to look at the level of a single cardiac cell. A cardiac cell can be in one of three states. It could be at rest, or in a depolarized state, or a repolarized state. At rest, every cardiac cell is maintaining a difference in electrical charge across its membrane. The inside is relatively negative, and the outside is relatively positive. And this is not a passive process. It costs energy to maintain. And this difference, this imbalance is everything. Because charge always follows one rule. It moves toward balance. So as long as that imbalance exists, the cell is ready for the next phase. When a cell is stimulated, certain ion channels on the membrane open. Ions rush across the membrane rapidly. The inside shifts from negative to positive. This is called depolarization. And depolarization is not just a change in charge. It is the signal that tells the cell, contract now. After activation, the cell cannot stay in that state. Ion channels open and close again in a certain sequence, pushing ions outside of the cell. The inside gradually returns to its resting negativity, and this is called repolarization. It is recovery, reset. And therefore, the cell returns to being ready to fire again. It returns to its rest. So the cycle is rest, then depolarization, then repolarization, then rest again. So the cells will go from activation, then they will go to recovery. And the truth is, without repolarization, there is no next heartbeat, no reset, no rhythm, no heartbeat continuity. Now let's zoom out one step back. The heart is not one cell. It is millions of connected cells. So what matters is not what one cell does in isolation, it is how that state spreads. So, when one cell depolarizes, it triggers its neighbor. That neighbor triggers the next. A wave of depolarization moves across the heart, followed by a wave of repolarization. This is how millions of individual cells become one coordinated contraction. Not chaos, a sequence. And here is the first bridge to what's coming. This wave has a direction. It has timing. It has magnitude. And if you place electrodes on the body, you can detect it from the outside. That is all the ECG is, a recording of these waves as seen from the surface. We'll come back to that later. First, we need to understand what still actually generates these waves. To understand the wave, we go one level deeper. The action potential. This is the precise sequence of ion movements that produces depolarization and repolarization inside a single cell. And the heart has two different types of cells with two different action potentials. Most working heart muscle, the walls of the atria and ventricles, use fast response cells. When stimulated, sodium channels open rapidly and sodium rushes in. The inside voltage shoots upward, a sharp, fast spike. This is depolarization in these cells. Then, over time, calcium channels open, calcium flows in slowly, sustaining the contracted state. This is called a plateau, a prolonged period where the cell stays activated. Finally, potassium flows out of the cell. The cell returns to its resting state and repolarization is completed. So this is it for the fast response cells. But the sinoatrial node and the AV node, the cells you already know, are different. They are slow response cells. They do not use fast sodium channels. They rely primarily on calcium. The result is a slower, more gradual depolarization. And this is not a flaw, it is a design. Because these cells are not built for force, they are built for timing. So the heart uses two different ion systems for two different jobs. Sodium, required for speed and synchronization in fast response cells, and calcium, required for rhythm and control for slow response cells. One drives the contraction, and the other sets the pace. Now here is where everything gains a second meaning. Those three ions we just discussed, sodium, calcium, and potassium, they are not just values on a lab report. They are the switches that determine whether a cell rests, fires, or resets. And the system is sensitive. A small shift in ion concentration doesn't produce a small effect. It changes timing and coordination across millions of cells simultaneously. That is where arrhythmias come from. Not from the muscle failing, from the electrical sequence breaking. And here is the implication that should change how you think about medications. Every drug that acts on the heart acts on ion movement. Some drugs slow the calcium channels. Others block sodium entry. Others affect how potassium leaves. When you change the ion behavior, you change depolarization, you change repolarization, you change the timing of the wave, and when the wave changes, what gets recorded from the surface changes too. The same is true for electrolyte disturbances. Elevated calcium, low potassium, sodium shifts. Each one disrupts the precise ion balance that the action potential depends on. The wave becomes different, and that difference gets recorded. I haven't said a single word about waves, intervals, or complexes in the ECG, but everything we've discussed, rest, repolarization, depolarization, conduction, ion balance, that is what produces them. Every mark on an ECG is just the surface expression of what we've been talking about. So when something disrupts the cellular level, a drug, an electrolyte, a disease, the ECG changes. Now that we understand what electricity actually is in the heart, we can finally learn what it looks like in the next video. Next time, we start reading. That's all. Thank you and see you in the next lecture.